Thank you all, and good afternoon. Delighted to be here. Thank you for the invitation and for the support. Many of you know me as the president of the Buck Institute, but today I'll be talking about actually the work that I still conduct in my lab. I still run a pretty large lab that's focused actually in large part on immune aging. So just a very quick way of introduction. We're all familiar, and we've heard from many speakers, the two main paradigms on which this field is working today is the idea that there are a number of aging-associated cellular dysfunction, the so-called hallmarks of aging, and along with this geroscience hypothesis, which is the idea that disturbance in the aging process is associated with an acceleration of the chronic disease of aging. And obviously the promise of this field is by targeting the modifications on the left, we will be able to interfere and prevent the chronic diseases of aging. So today I'm going to be focusing on senescence, and I want to dedicate this talk to actually my colleague, Judy Campisi, who left us way too early this year. Judy was a true pioneer in the whole field of aging research, and we miss her obviously very much. I think the whole field misses her. Much of our work at the Institute has actually been focused on understanding the drivers and the mechanism of senescence, and I'm going to be talking about this in the context of the immune system. So what is senescence? If you have not heard before, this is a cellular process, a cellular program that is activated by a whole number of factors, including what we call replicative senescence, which is triggered by telomere erosion, but also other mechanisms such as inappropriate oncogene expression or a variety of stress such as irradiation. These lead to a complete reprogramming of the cell, and in particular to the secretion of something that Judy and her colleagues identified at the Buck, the so-called SASP, senescence-associated secretory phenotypes, which is a series of pro-inflammatory molecules. These molecules have profound effect on the organism. First, the first loop that you see going on the top is the activation of the immune system, which recognizes these cells and eliminates them over time. The second one is what we call the paracrine, which is, I think, even more important. It's the idea that these pro-inflammatory molecules can induce secondary senescence in other cell types. And I'll show you in a few minutes why I think this is really, truly important. So this is so-called secondary senescence or bystander senescence. And obviously, the thinking is that this is one of the major contributor to a whole class of abnormalities associated with the immune system that we refer to as chronic inflammation or inflammaging. Now, why do I think this is really important? There are two stories that I'm highlighting here, recently published in 2020 and 2021, both showing the same thing, is that a primary lesion in the immune system, senescence by creating dysfunctional mitochondria only in the immune system, or a defect in DNA damage repair in the immune system only, these primary abnormalities in the immune system induce secondary senescence in non-immune organ. And this is shown, I'll point to you. Do I have a pointer here? I guess no pointer. I'll point to the slide here on the bottom right, which shows a whole series of tissues, aorta, kidney, lung, liver, spleen, all showing secondary senescence occurring from a primary lesion that happens in the immune system. Now, the reason for this is, why this is important is the immune system is the only distributed organ. If you think about it, the whole group of immune cells represents an organ which is distributed across the whole organism. And so abnormalities in that compartment can have dominant effect on other tissues. And so this is the basic foundation of why we think the immune system represents a unique target for interventions and for understanding the process of aging. And we've heard the talk from Greg Fahey, who is actually addressing a part of immune aging. Now, some of the key questions that this is a project that actually started only about 5 years in my lab, and the focus of the questions that we're trying to answer are here. First, is there immune senescence in humans? Can we actually detect it? Can we measure it? Does it change with age? Are these cells actually truly pathogenic? And can we target them for elimination and therefore really kill one of the primary, what we think is the primary driver of aging in humans? Now, the immune system is obviously very complicated, and I won't go into this slide because of the complexity. The only thing that I want you to know is that there are two broad, all immune cells originate from a central hematopoietic stem cell, which is located in the bone marrow. And the immune system itself divides in two different parts, the so-called adaptive immune system, which is your T cells, and B cells, which is an immune system that is able to be educated in contact with foreign pathogens, for example. There's also a sort of a myeloid immune system, which would be your monocytes, your eosinophils, neutrophils, and so on, which is a more proximal early defense system. Now, this picture looks complicated, but I'll show you it is getting a lot more complicated the more we study it. And we've heard also from Greg just before the intermission, that one of the hallmarks of aging in the immune system is thymic involution. So the thymic is a key place for maturation and education of immune T cells, which is part of the adaptive immune response. And we know that by the time you reach 50, most people will have only a small remnant of this thymus. And so this has obviously enormous implications, because once you reach 50, you are left with a pool of immune cells with which you have to deal for the rest of your life. And so here, without getting into too much technical details, I think it's important for you to understand that these immune cells, these T cells, are generated in the thymus. They become in the thymus educated in recognizing the self from the non-self, and a breakage in that education process is what leads to eventually autoimmunity, where the immune system will turn against you. Most of them actually do well, and actually they are targeted uniquely to pathogens. Now, another reason why the immune system is important has been really highlighted to us in a very acute way recently, where the COVID pandemic showed that an incredible dependency of your chronological age and your risk of dying. Actually, the risk of dying can be up to 70-fold higher depending on your age. Now, this has been illustrated with COVID-19, but it has forced many people to go back and to realize that the same goes for influenza, the same goes for RSV, respiratory syncytial virus, and many other infections. So with COVID, we just saw the tip of the iceberg. It's clear, and we've shown it, I think Greg showed us another slide where infectious diseases really kill disproportionately the aged. And so this is really one of these bottlenecks and rate-limiting factors that prevents us from living old and well. Now, going back to the naive T cells, which is the precursor that originates in the thymus, eventually it migrates out of the thymus and populates the peripheral lymphoid organs. And as these cells become educated or recognize their foreign antigens, they will undergo a number of transitions. They will become early memory, central memory, effector memory, and eventually terminally differentiated T cells. Now, so we did a very simple experiment to start this. We were able to purchase and to obtain in-house a new machine called the Cytek Aurora Spectral Flow Cytometer, which is a sort of cutting-edge flow cytometry. This is technology that uses fluorophores that we can attach to specific antibodies and recognize protein at the surface of immune cells. We started with a very small population, so this allows you to stain for 38 colors versus the standard flow cytometry, which uses 5 to 6 colors. So I'll just ask you to imagine in your mind a matrix of 38 dimensions. This technology now allows us to detect more than 650 distinct populations in peripheral blood. Therefore, something that used to be somewhat simple on the slide that I showed you is becoming incredibly complicated. And along with this complexity, I think we hope we'll gain some much deeper understanding of what's happening in the immune system as we age. I should say also one of the incredible things that's exciting about the immune system is that this is the only compartment that we have access to on a daily basis. Nothing easier than to draw blood and to conduct this type of analysis. So we think there's a really unique opportunity, both in terms of the importance of the organ for aging, but also its accessibility, not only for measurement, but also for therapeutic interventions. So this initial study that we did used about 40 different individuals of different ages and different genders. And I'm showing you on the left what we call a dimensionality reduction plot, or UMAP plot, which takes all of this data from these 38 dimensions, and we use 4 different panels, which each have 38 dimensions, allows you to reduce this into 2-dimensional plots, which identifies a whole series of cell types. And you can recognize in here your CD8 T cells, your NK cells, and so on. And when looking at the effect of age, we see some things that have been reported before. So this is sort of make you feel comfortable with the quality of the data that we generate. For example, monocytes increase during aging. CD8 T cells decrease during aging. So do gamma delta T cells. Both of them are dependent on a part of the adaptive immune system. We can also do the same analysis, not just focusing on the CD8 T cells that I discussed earlier. And here again, we find actually an increasing complexity in contrast to what was known previously. You can see even the CD8 T cells can resolve into close to 14 different populations. And we see the same thing now at every level of the immune system. And here the pictures become even more acutely evident in terms of the effect of aging. Greg told us about the decrease in naive T cells during aging, and you can see how steep this decrease. We know about thymic involution. This is where the naive T cells are made. You can see by the time you reach 80, you almost do not have any more naive T cells. We've also been able to identify some novel cell types, and I won't talk about this today, but these central memory CXCR3- cells are essentially absent when you are young, and we can see them increasing dramatically, whereas they can represent up to 40% of your total CD8 T cells. So we're doing a lot of work on these novel central memory T cells. They have, for the aficionados, they have a Th2-like phenotypes, which would probably make them help explain some of the increasing incidence of asthma and other immune complications that occur during aging. Now, moving on to senescence, what is the role of senescence in these processes? Can we actually measure senescence in the immune systems? And again, here I'm pulling a paper from Judy Campisi, showing you, she was the first one to identify a specific biomarker for senescent cells, a so-called senescence-associated beta-galactosidase. And the way this works is beta-galactosidase is an enzyme which is expressed in lysosomes, and if you feed it this hybrid molecule called galactose indigo monomer, the galactosidase will cleave it, the indigo will dimerize, and will generate a blue marker that you can detect by simple staining. So this test was really instrumental for many things in the field, opened up the ability of people to measure what is senescence and to regulate it. And interestingly, there's a derivative of this assay that we can use on immune cells. And this is illustrated here. This is one of them called FDG. There are others, Spider-Gal, and so on. Again, the same principle. In this case, you have a fluorescent molecule, FITC, which is coupled at both sides with galactose when galactosidase is present. So the fluorescence is quenched under these conditions. If you cleave it with the galactosidase, you generate FITC, which can be detected by flow cytometry. So you can now couple this type of analysis with all of the high-resolution flow cytometry that I've discussed earlier, and this is shown here. So we're comparing here a young, which is below 40, and older donor above 60, and so to group them. And you can see when we compare each of these different cell types, these immune cell types, going from the naive all the way to the terminally differentiated, the EMBRA, you can see that the red dots keep increasing, both in CD4 and in CD8 T cells. Both cell types have the same type of differentiation process in the thymus and so on. And again, here, dramatic numbers. If you look at the senescence field, typically the fraction of cells that are senescent in most tissue as a function of age rarely goes about 2 or 3%. In this case, we see clearly in the more terminally differentiated, up to 40% of these cells becoming senescent. So it could be, we still haven't resolved this clearly, and I'm going to try to convince you in the next few slides that these cells are indeed senescent. But this would have enormous implications in terms of what the role of the immune system. If 40% of your effector memory T cells, so-called EM, are becoming senescent, this would be distributed across the whole organism with the potential of really creating some significant problems. What's interesting also is that these cells go through their differentiation process. They divide more and more. And so there are two, but we see it in the naive T cells as well. So it's not only in the terminally differentiated. Now, one way to actually determine whether these cells are truly senescent is to conduct a deep molecular phenotyping. We did this based on the so-called Sen-Mayo data set, which was generated by a group at Mayo Clinic, where they identified a core set of genes that appear to be co-regulated in senescence. One of the problems of senescence is that it is a varied phenotype depending on the cell type. So there are no single markers that define senescence in any tissue. Not P21, not P16, the ones that everyone associates. You need a constellation of change in gene expression and phenotype. And so the Sen-Mayo data set tries to address it. So we use this data in our experiment, purified either CD4 or CD8 central memory T cells or effector memory T cells, and looked at the differential expression of genes in the beta-gal positive and beta-gal negative, the red dots versus the other ones. And what we were able to show is that there is a very strong overlap, which is shown in yellow. Every time you see a yellow band, it just tells you that that particular gene is overexpressed. And so the whole gene set here is Sen-Mayo. You can see many of them actually are overexpressed in the SA beta-gal positive, just fulfilling the first prediction that these cells are indeed looking like senescence. We've done a whole series of other markers that I'm showing you here, including looking at the lysosomal content of these SA beta-gal low versus SA beta-gal high. And you can see here using a marker called lysotracker red. If you look at the top versus the bottom, you can see a very large enrichment in the lysosomes here on the middle panel. And we can quantify this. So this is, again, something that's known, but it makes us happy to see that this is fitting in these cells. We also see a decrease in, let me see, I'm blank, in P62, which is a marker for autophagy. And here again, if you look, the P62 is actually dependent, or LC3 are dependent on autophagy. So if autophagy is inhibited, which we know it is in senescent cells, you can see a large increase in the staining, as I'm showing you on the 3 cells at the bottom, or the quantification. We see also chromatin fragmentation, which is a really interesting phenotype. During senescence, the chromatin from the nucleus gets excised, goes into the cytoplasm, and along with the DNA, and this triggers a DNA damage response, which we can detect using a modified chromatin called gamma H2AX. And you can see in the SA beta-gal high, you can see this incredible punctate pattern around the nucleus, which is indicative of this chromatin fragment. Now, when you see this, it's clear to me that no one should ever think about reversing senescence. Senescence is a process that we should really fight to eliminate. These cells obviously have massive DNA damage, which is not reversible. This DNA damage is not going to go into the nucleus at the right place. So there's, I've heard about the idea, why don't we just revert the senescence process? Senescence occurs for a protection mechanism, and so when you see this type of picture, you realize that we're better not to touch them in terms of allowing them to survive. Now, there's another marker that has been of interest to people in the field of aging is lipofuscin. For those of you who are old enough, you probably have noticed some of these aging spots that you can see mostly on your hands or on your face. These are aggregates of lipids and proteins cross-linked called lipofuscin. They are thought to be an end product of dysfunction of many things in the aging process. And obviously there's concerns and interest in trying to remove them. Now we think, and I hope to convince you that we think that lipofuscin accumulation is a marker of senescence as well, at least in the immune system. So one of the beauty of lipofuscin is that it is naturally fluorescent. So you can actually, at a certain excitation wavelength, you can actually detect it. And so we use our high-resolution flow cytometer to do this. This is something you cannot do with a standard flow cytometer. And this allowed us also to compare, again, the young and the old, or the SA beta gal positive and SA beta gal negative. The first thing I'm showing you here is, as a function of age, we can detect a clear picture of lipofuscin in T cells as they develop. Again, look at the red dots on the right side. You can see a clear increase. Interestingly, when we use in vitro experiment and we treat cells either with chloroquine, which is an inducer of lysosomal dysfunction. Now, the reason why we think that these are two, SA beta gal and lipofuscins are two markers of lysosomal stress, essentially. They're measuring the same thing using two different tools, and they are largely congruent. If you look at the FACS plots on the left, you can see that there is a line, you can draw a line diagonally across between lipofuscin and lysosensor blue, which is a marker of lysosomal function. So we think, again, if we think about lipofuscin, we think about it under the same mechanism and the same terms as lysosomal dysfunction associated mostly with senescence. And so this obviously somewhat solves the problem in a way that I do not think that lipofuscin is an entity that can be pursued by itself. If we solve the senescence problem in the immune system and probably in the skin and other tissues, we should take care of the lipofuscin as well. Now, one of the interesting aspects that came out of this work is the idea, if we see this progressive increased senescence in the immune system as cells differentiate and as we age, can we actually measure this using some of the epigenetic clocks? And so one of the simple experiments that was conducted by a graduate student in the lab was to take 7 donors, again, aged 30 to 77, and to purify using sorting. Now, flow cytometry does not only allow you to detect the cells, it allows you to sort them individually based on whatever marker you choose. So we can sort the naive T cells to homogeneity, to 99% purity, and then conducted a DNA methylation analysis, asking a very simple question. When we isolate the cells from someone who's 50 years old, do all these cells look the same in terms of age? And my initial assumption was that they would be, but I've just showed you that they don't have the same level of senescence. Can we actually measure this? And so this is the data that actually Greg alluded to that really, when I first saw it, I had a hard time to believe it. So if you take your naive T cell, central memory T cell, effector memory T cell, TEMRA, and you measure a whole variety of clocks, and we've done this with all of them, going from Horvath, the skin and blood from Horvath, or phenoagents. We've done all of them. It will be in the paper that's coming out. We find close to 25-year difference in epigenetic age between these cells, meaning that the naive T cells are on average 15 years younger than your, actually 25 years younger than your effector memory T cells. Now, the implication of this that I have pointed to Greg and his colleagues is that if you're shifting the balance, remember I told you that during aging, the naive T cells are going down, so obviously you are, and the effector memory are going up. This is going to be meaning that you're going to have the impression that the person is aging or reversing aging. And obviously I don't think this is a form of aging, but this is not aging in these cells. This is what, to try to remedy this, we have advanced the concept of intrinsic and extrinsic aging. Okay, so each cell itself can have its own age, but in addition, changes in relative proportion of these cells in the blood can give you the impression of aging or not. And I think this is something that we should always keep in mind. Now, to try to look at the data in another way, If you take only the naive T cells, look at the blue line, we find that first at the first point at age 30, you can see again the naive T cells are younger than the central memory, younger than the effector memory, and younger yet than the TEMRA. Now, so that's what I just showed you. What's interesting is that in addition to this, there's a signature of aging in the naive T cells themselves, and this is what we call intrinsic aging. So these cells do age. In addition, there are shifts in the populations. Now, to try to eliminate this problem, we created a new clock called Intrin clock, which is coming out, and based on a large data set. And we did this by eliminating up for the aficionados all of the CPGs that are linked to T-cell differentiation. And so this new clock, as you can see up, we call it Intrin clock. It's available from TrueAge right now, True Diagnostics, and we're planning to share it with others in the futures as well. You can see that the clock is very highly predictive in terms of predicting age, not only in somatic tissues, but in blood, brain, and other tissues, including the saliva. I'm always puzzled and intrigued by the semen. You can see that semen is the one tissue that actually shows no aging. And it actually has an age of 16 years old, which is really interesting, puberty. So there's something really interesting about the biology that we don't fully understand. Now, interesting, this Intrin clock does not vary during the differentiation from naive to central memory. You can see how it stays pretty constant, whereas the Horvath clock changes. And again, when we look at individual donors, they actually are pretty stable. Interestingly, this clock still speaks an aging language in a way that if we reprogram these cells shown on the left, you can see that using the Yamanaka factor, you can see a progressive regression all the way to an age 0, which I think is pretty amazing. The other clocks regress it, but not quite as much. And more interesting for us, the clock also measures senescence in a way that we do not fully understand. So here's on the right, you can see a number of passages. This is a replicative model senescence where the cells progressively become senescent, and the clock really is able to measure this. So perhaps intrinsic aging is actually measuring, again, the proportion of senescent cells in a given population. So this was going to be my summary, but I'm going to skip it for 1 second to show you another thing related to immune aging, which is a clinical trial that we recently completed in collaboration with Timeline or Amazentis using urolithin A, which some of you might know is a natural product, which is showing remarkable effects. And so we conducted a collaboration with them and the George Speyer House in Frankfurt, where we administered urolithin A to individuals for a 1-month period. And so our lab and our effort was to try to immunophenotype these donors. And what's pretty remarkable is within 1 month of treatment, and which I was against, to be totally honest, about the idea of doing such a short time treatment. Even within 1 month of treatment, we see an increase in the number of naive T cells. This is shown here in the red bar. It's not huge, but it's real and it's highly statistically significant. And my anticipation is as we do this in the future, this will continue to rise. So this is the type of, again, interventions that could really become very promising in the future. And I want to close on this by thanking Forever Healthy, who helped, and the organization helped us actually fund a significant part of this work. But also want to highlight Michael Antonoff Foundation, Stephen and Mary Reed, the NIH, and the part of my lab that is focusing right now on immune aging. And I'll stop here, and I'll be happy to take your questions.